In the second recording, we will continue to discuss classification of mutations, and we will start with another way that mutations are classified, and it is based on their phenotypic effect. In the most general way, we can actually distinguish a mutation on the basis of its phenotype as compared with the wild type. If we do so, we can classify mutation into two major categories: forward mutations and reverse mutations. Forward mutation change wild type into mutant type, while reverse mutation change that mutant type back into wild type. Now, in other terms, how to describe the effect of base substitutions on the phenotype are outlined right down here in red. We can distinguish missense mutations, silent mutations, neutral, and nonsense. Missense mutation is a base substitution that results in different amino acid being incorporated in the polypeptide, so it changes primary structure of the protein. Silent mutation actually change the codon. The mutation changes codon, however, it's a different codon for the same amino acid, so the different DNA sequence will result in the same polypeptide change, so amino acid incorporated in the polypeptide change will not be changed. Neutral mutation is a missense mutation that alters amino acid sequence of the protein, however, it does not change its function. How is it possible? You remember, amino acids are distinguished by functional groups. Now, if the amino acid which is replacing the original due to change of codon has the similar size as the original, has similar charge, or is doesn't have charge, so if it's similar to original one and it does not alter secondary and tertiary structure, basically it does not change its function of the proteins. Neutral mutations are very important to tracing to follow the evolution, and you will talk more about those in evolution class. The last one are nonsense mutations. Nonsense mutation changes sense codon into nonsense. What does it tell you? That the codon that is a change, the mutation actually introduced premature stop codon. So did you notice in the previous slide that base substitution, which is one class of the mutations, can actually have different phenotypical effect? Phenotype can be different. Here is one example. You have here mRNA codon and mRNA, which is UCA. Now this codon encodes serine. So in wild type polypeptide there is serine. If there is missense mutation, basically what happens is G in the template is mutated into A, and basically it changed the coding sequence from UCA to UUA, and now instead of serine, leucine is incorporated. So basically base substitution caused missense mutation, change in amino acid. Now, base substitution can also result in nonsense mutation. It's outlined right here. If here instead of G is incorporated into the template DNA thymine, this will change UCA codon to UAA codon, which is stop codon, which would trigger premature termination of protein synthesis, so it is nonsense mutation. And because there is redundancy of the genetic code and different codons can specify the same amino acid, we can also, base substitution can also trigger silent mutation. Right here, UCA codon is changed to UCG, and both of them encode serine, so it is a silent mutation. So be aware that this classification is not strict, it's relative, and basically once we classify the mutation on the molecular level, such as base substitution, this base substitution can have different phenotypic effects, so those mutations can be classified under different phenotypic categories. When we discuss mutations, we often connect them to loss of function, that mutation trigger dysfunctional proteins. This is only one group of mutations which we call loss of function mutations. They trigger complete or partial absence of normal protein functions, and they are frequently recessive. We discuss some of those mutations, such as cystic fibrosis, for example. Opposite to loss of function mutations are gain of function mutations. This mutation gives the cell or tissue entirely new trait, or the trait which normally is physiological in certain tissue appears in new tissue, in an inappropriate tissue. For example, growth factor receptor. Can you imagine if growth factor receptor to VEGF appears, let's say, in brain tissue, and now they start to respond to growth factor and proliferate, it can have deteriorating effect. In addition to loss and gain of function mutation, you can recognize also so-called conditional mutations. These mutations are only expressed under certain conditions. One of the best examples are a mutation which are triggering temperature sensitive condition. This temperature sensitive mutation basically do not manifest in an individual who carries them if that individual is exposed to normal range of temperatures. However, if it is exposed, he or she is exposed to high temperature, this can be deteriorating. It can have very serious effect. And the last one are lethal mutations. Lethal mutation cause premature death, very often even during embryogenic development. You recall we discussed lethal mutations when we talk about the exemption from Mendelian laws. Lethal mutation can alter expected phenotypic ratios of the offsprings. A very interesting group of mutations are suppressor mutations. They can be intragenic or intergenic. Suppressor mutation actually trigger changes of mutated phenotype into wild type, back to wild type phenotype. However, they are not reversed mutations. They do not reverse original mutation. What they do, they introduce additional second mutation that actually hides or suppresses the effect of the first mutation. So let's look at this first on intragenic. Intragenic means that it is within the same gene. And what happened here, let's say we have codon UAA, which encodes leucine. Now, a missense mutation which changed in the template time into adenine changes UA codon to UUU, which encodes phenylalanine. So there is a missense. Leucine is changed to phenylalanine. Now there is a second mutation introduced at different site. You recall the first mutation was a third nucleotide in the template. Now the first nucleotide is changed, is mutated from adenine to guanine, which changes codon to CUU, and CUU similarly to UUA encodes leucine. So suppressor mutation is not reverse mutation. It does not change back the mutated nucleotide. If it was reverse mutations, this adenine would be reverse mutated back to thymine. Suppressor mutation introduces second mutation, which actually reverses the phenotype back to the original wild type. This is an example of intragenic mutation. In the next slide, we will discuss intergenic suppressor mutation. An intergenic suppressor mutation occurs in gene other than the one bearing the original mutation. If it's intragenic, it is in the same gene. If it is intergenic, it is in different gene. And here is an example of intergenic mutation. Let's say in the first picture right here, you have normal synthesis where basically leucine is incorporated in protein, and there is a codon UUG which actually interacts with anticodon AAC. Now, mutation in template triggers changes of original UUG codon to UAG, and we know that UAG is stop codon, which result in shortened non-functional protein because this is nonsense mutation, premature termination. Now, where the intergenic suppressor takes place? In the gene which encodes tRNA. Normally, let's say tRNA which carries tyrosine would have anticodon AUA. Now, this anticodon might change to AUC, and now AUC here will actually recognize UAG and will not actually stop, but will translate. Translation will continue. You remember there is normally no tRNA which would recognize stop codon. However, this mutation introduces in second gene, in gene for tRNA mutation, which now carries anticodon which would recognize that mutated codon. So the mutations happen in two different genes. That's why they are intergenic. So in these slides, you have a table which summarizes all types of mutation we discussed in this and previous recordings, and also gives you a definition. So what I advise you is to use this for review. Take piece of paper and cover definition part and read the type of mutation and see whether you can define what kind of mutation it is. It's a good way how you can actually test yourself. In the last two slides of this recording, we will discuss mutation rate and adaptive mutations. So what is mutation rate and why do we care about? So mutation rate is the frequency with which a wild type allele at a locus changes into mutant allele. Now, how do we measure these mutations? We can measure them as the number of mutations per particular unit, either per cell division, per gamete, or per rounds of replications. And of course, we care about rate of mutation because more mutations which are not repaired may result in less functional proteins and altering in normal functions. So, what affects the mutation rates? There are several factors. Firstly, the frequency with which mutation occurs. We know that there are spontaneous mutations which can occur with higher frequency in some DNA regions than in another, and plus mutation can be altered by environmental factors such as chemicals or radiations. The second factor that influences the mutation rate is probability that when mutation happen, it will be repaired. Most of changes are actually repaired if DNA repair mechanisms are functioning normally. However, some mutations are increasing over the time, and if the mutation affects the genes that encodes component of the replication machinery or DNA repair enzymes, the frequency rate of mutation will increase. The third factor is the probability that a mutation will be detected. You remember we have mutations such as neutral mutation or silent mutation, which actually do not show in phenotypes. They do not cause phenotypic effect, so they are much more difficult to detect. Now, are mutation rates the same? No, they vary between species, they vary within the same species between genes. Spontaneous mutations are usually rare. In bacteria, it might be 1 to 100 mutation per 10 billion cells, while in eukaryotic cells, it varies between 1 to 10 mutations per million gametes. You see, they are actually recorded per different biological units, which makes it more difficult to compare it. Now, even if you look at the genes in the same organism, let's say in eukaryote, there are differences. Certain regions of DNA in humans, for example, or in other organisms, mutates with higher frequencies than in others, and those we call hotspots for mutations. Usually DNA which is associated with nucleosomes, genes which are found in this part of DNA, are less exposed to mutagen and usually have a lower frequency of mutations. It is estimated that in humans, in about 1 time 10 to 8 mutation per base pair per generation occurs. So let's talk about adaptive mutation. We know that genetic variation is necessary for evolution. Without genetic variation, natural selection and evolution cannot happen. Now, new genetic variation arises primarily through mutations during many generations. For many years, we consider or we assume that the mutation arises randomly, and basically if some of this randomly arised mutation gives the individual advantage, it would be selected. This happened, for example, if you are interested in viruses like HIV. When HIV replicates, reverse transcriptase introduce a lot of errors, so there are many mutations. However, they are less fit than wild type, so normally wild type prevail. However, if you suppress wild type by drugs, some of those mutations can actually support resistance or encodes resistance to the drug, so now this mutation is selected. That's what the view of mutation, of variation, genetic variation created by spontaneous mutation was. However, recent evidence, some evidence suggests that under particular conditions, which actually are very stressful environment, may trigger increased rates of mutations, and these mutations provide adaptation necessary to survive. So basically, stressful environment can induce more mutation, and these mutations which are necessary to survive we call adaptive mutation.